Okay, hello and welcome to the metabolic pathways topic. It's part of the living cells unit of the higher human course. Now, I'm going to try and get as much of this into about 10 minutes as possible, so I'm going to take you quite quickly through it. Now, you remember that this video can be used in that sense where you can fast forward, rewind, pause, maybe re-watch bits again, so don't worry if you feel like I'm moving too fast, because you're in complete control of the speed I move at. First thing I'm going to talk about is what an actual metabolic pathway is. What we have previously understood at National 5 level is that an enzyme-controlled reaction consists of a substrate being acted upon by an enzyme to give us a product. Now, in real life, in real cells, that is rarely the case. What's more often the case is that a series of enzyme-controlled reactions is how we get our product. So we start off with a substrate. That would then be acted upon by an enzyme to give us an intermediate. Yeah. Maybe another intermediate is formed as well by another reaction, also controlled by another enzyme. Remember, enzymes being specific in their action. If the enzyme 1 does that reaction, then it can't be enzyme 1 that does that reaction. This is what we learned from National 5, of course. An intermediate B might then form the product when acted upon by a third enzyme. It's also worthwhile at this point considering the knowledge that we can tie together from other topics from the higher course. Enzyme 1, 2 and 3 are all proteins, are therefore would be coded for by 3 different genes. And again, tying together knowledge that we've already gained through the course, through Unit 1 so far, we do understand that mutations can take place. So if your mutation took place in gene 2, which meant that enzyme 2 was not produced, then actually what would start to happen is this pathway would break down because this reaction wouldn't be able to happen. So what would happen as a result of gene 2 being mutated to the point enzyme 2 couldn't be produced and this reaction couldn't take place is that the intermediate A would start to build up. So we would get an accumulation of intermediate A in the cell. Eventually, of course, intermediate B would run out because there's none being formed. So intermediate B would run out, and if there's no intermediate B for enzyme 3 to act on, there would be no product. So the whole pathway breaks down. Now the cell can do a couple of things in response to that. It's worthwhile understanding that reactions quite often, not always, but quite often are reversible. And as a result of that, when a product builds up, if the reaction can't go in that direction, then what will actually happen is the reaction might reverse. So a solution to this intermediate A building up might be that the cell would reverse its reaction. And actually the direction of some reactions is completely controlled by the concentration of the reactant and the product. So if there's more reactant than product, the reaction goes that way. If there's more product than reactant, the reaction goes back the way. Now that doesn't always happen. Often reactions are not reversible and therefore that's not a solution. So in that particular case, the cell still has another get around, quite literally, a get around not having enzyme 2 working properly. What it will do is it will use and employ another enzyme. In this case, enzyme 4. And what enzyme 4 might do is it might act on intermediate A to form intermediate C. And intermediate C can be acted upon by another enzyme, enzyme 5, to give us intermediate B, which means we don't need that faulty enzyme. That's what we call an alternative pathway. Okay, so some reactions can be reversible and also alternative pathways can be employed. Both of them get around the fact that an enzyme might not be working properly. So those two are worth considering as solutions to that reaction, to a reaction not operating properly. And remember, the direction of the reaction is controlled, and the rate of reaction actually, so direction and rate of reaction are both controlled by the concentrations of the reactant and product. A bit like the fusion in a sense. So if you've got a very, very big concentration gradient between here and here, then the reaction would go quite quickly backwards. On the other hand, if the concentration gradient means there's a very high concentration here and very low concentration there, the reaction would go forwards quite quickly. The reaction slows down as the reactant and product concentrations become similar. Probably time to move on to two new words that you will learn. We understood at National 5 level that enzymes controlled two types of reaction. One of them we called synthesis, which involved the build-up of small substrates to large products. Another one was called degradation, where large substrates were broken down into small products. Now that wasn't not true, but there